Hello everyone, welcome back to one more video session. In this session, I'm going to explain about one gene, one enzyme hypothesis proposed by Beadle and Totten. Very important for your competitive examinations. That's what. It is not required for board. So we just write one gene, one enzyme. It's very simple. According to Beadle and Totten, they say that the single gene produces only one protein. That is nothing but enzyme. Okay, so one gene, one enzyme. Hypothesis proposed by, we just write the name of the scientist, Beadle and Totten. Please remember the names of Beadle and Totten. They proposed one gene, one enzyme hypothesis. Very important. Even you get the medical seat, you have to study this particular concept. So please remember this. This is the basic. And for this, actually, according to them, they says that the a single gene will produce only one type of protein. Okay. So if you take any type of living organism, what kind of proteins will be there? Basically, for the survival we need enzymes, because enzymes play an important role in the metabolism process, correct? So these enzymes are the important proteins that is required for our survival, correct? Okay, other than that there are so many proteins like structural proteins, even receptors like present on the cell membrane, we know about it. Okay, but here one gene, one enzyme hypothesis was actually, it was targeted only on the enzyme's activity within one organism. They selected a type of fungus called as neurospora. We just write neurospora. I'm not following the binomial nomenclature rules. Neurospora crassa, a type of fungi. Okay, they just selected this for the experiment to prove that one gene will synthesize only one protein, or one gene will synthesize only one enzyme. According to them, all enzymes are proteinaceous. Okay, we just write all the enzymes are proteinaceous. First, before we see their experiment, let us just understand the basics. So I'm going to draw a single stranded DNA. Let us assume that it is the template DNA strand. Okay, so this is our template DNA strand. Let me just stretch it, but I am using different colors. So it should be noted that within the same template DNA, or not same, like it is the like long DNA, and this is now one template DNA strand, and this is another template DNA strands. They are present at regular intervals. Okay, so this particular DNA template strand will produce one mRNA, that is nothing but transcription. That mRNA will produce a type of protein. So this is now one gene. So we will name it. The white color one is a single gene. So this is gene 1. Let us just assume. After certain intervals, let us say like there are some useless bases will be there, and that cannot be called as gene. But after that useless bases, you get the purple color segment. So that is gene 2, because it is going to produce the protein. It could be enzyme. It could be any type of protein. Okay. So I am using the pink color. So this is now gene 3. Okay, it is understood. Gene 3. Okay, so these genes are obviously they will undergo transcription. Here also, this also undergoes transcription. This also undergoes transcription. What they are going to produce? They are going to produce the mRNA. Correct. So this is one mRNA of gene 1. So we use purple color. This is another mRNA of gene 2, and we use the pink color. So this is another mRNA of gene 3. Okay, they could be produced simultaneously or maybe at different time. Doesn't matter actually. Okay, it's understood. Now this particular mRNA will now undergo translation and will produce a polypeptide chain. So I think not necessary. Okay, so I'm using pink color. But you just understand this is the product of gene 1. Okay, so this is now the polypeptide chain, the amino acid sequence of the gene 2, that is another protein enzyme, and here the enzyme 3. So we will name it. This is enzyme 1, this is enzyme 2, this is enzyme 3. Right? Hope you are getting my point. Okay, so gene 1 will not produce enzyme 3. Gene 2 will not produce enzyme 1. It is understood. They are very specific. Gene 1 will produce only enzyme 1. So what are enzymes? Enzymes are nothing but these are the proteins. They are very very important for the metabolic reactions. We know about enzymes. Okay, so here let me just tell how exactly they prove that single gene will produce only a single enzyme or single protein. This is the thing, because if you look into the prokaryote or the eukaryote, you know that when I was explaining, I said like a polycystonic gene in prokaryotes, whereas monocystonic gene in eukaryotes. In eukaryotes, that monocystonic gene, the single gene will produce only one protein. In the polycystonic gene, a single gene will produce multiple proteins, correct? Okay, but according to them, no. According to them, it is entirely different. Okay, so we see that. So they grew the neurospora, that is the fungi, in a petri plate containing a simple medium, because they selected neurospora because it can be grown in such a simple medium. Like it is not required to put like very expensive medium in the laboratory condition. So it could be easy for them to grow them in a simple medium. So let me just write simple medium, petri plate containing simple medium, and now I am using pink color. So these are all the neurospora, not exactly the actual structure of the neurospora. Okay, so just for our understanding, so these pink dots or circles are nothing but the neurospora. We all know that all living organisms require nutrition for their survival or for growth, reproduction, whatever it is. Okay, so they have to take up the nutrients. Yes, similar to us. Now I am going to draw one neurospora, that is like I will take this neurospora and I will draw it here. Okay, so this is now the nutrient N. N is now taken inside by the neurospora. So this N should be converted into certain chemicals because it is a metabolic pathway, correct? So metabolism. What is metabolism? It is nothing but the process of breaking complex organic molecules into simple organic molecules to synthesize ATP. Ultimately, what is required? ATP should be synthesized. For this process, you will see a lot of chemical reactions. Ultimately, ATP synthesized for conducting this lot of chemical reactions. Because if you look into Krebs cycle or glycolysis, obviously you have seen set of what you call reactions, correct? Okay, you see different types of chemicals in the different steps. The conversion of these chemicals are mainly facilitated or influenced by enzymatic activity. That means a particular type of enzyme is required for the conversion of these chemicals, and ultimately a product is produced. Okay, so during this chemical process, you know that somewhere else the ATP will be liberated, correct? It comes out. That is actually the thing. The same way here, the nutrition is now already like taken up by this neurospora, and this nutrition is converted into ornithine. Now we just write here, somewhere else here. Okay, I write ornithine. The first chemical, that is ornithine in the neurospora. Okay, so for the conversion of a simple nutrient into ornithine, it requires one enzyme, that is enzyme 1. They observed that a particular gene produces that enzyme, and this enzyme converts this simple nutrient into ornithine. Note this point. And after that, ornithine cannot liberate ATP. Again, it has to undergo set of chemical reaction. That means actually, it has to be simpler later. This is complex. Now, it has to be converted into another type of amino acid. Okay, so that is citrulline, not amino acid. It's another chemical. Sorry. Okay, citrulline. Okay, citrulline. It's like ornithine got converted into citrulline. For this conversion, it requires another enzyme, that is enzyme 2. Got it? Simple nutrient is converted into ornithine by the activity of enzyme 1, catalytic activity. So that means it is understood that gene 1 plays an important role in the conversion of simple nutrient into ornithine. Note this point. Then ornithine again got converted into citrulline by the activity of gene 2 because it produces enzyme 2. Got it? You may ask me like how did they know about it? Simple. If we just put X-ray here, okay. So X-ray means we know that it is mutation. You can induce mutation in this segment of the DNA. So if mutation has occurred in gene 2, what's going to happen? It will not produce enzyme 2. What is the result? Ornithine will not get converted into citrulline. So once you extract the chemical which is present within this neurospora, after some days, you get only ornithine. You will not get citrulline. Hope you understood. This is the method. Okay. Again, citrulline. By the activity of enzyme 3, that is by gene 3, the product of gene 3, enzyme 3, it got converted into arginine. Okay, remember it. So when they observed that when they extracted arginine from the neurospora, it was understood that all the three genes are functional and all the three genes are actually produced each enzyme. So enzyme 1, 2 and 3, and ultimately ornithine, citrulline and arginine, finally arginine got produced. Okay, so this is the thing, arginine was produced. Power cut, don't worry. Okay, hope it's visible to you. And this okay. And finally they named the arginine producing neurospora as prototropes, not phototropes, prototropes. Please note this point, you may get this question for your need. Prototropes are nothing but the neurospora which can produce arginine. It is understood that if arginine is produced, all the three genes are or enzymes are functional. Got it? It is not mutant, it is natural, normal. Please note this one. Prototropes means it is natural or normal neurospora. Now, they, very simple, they just mutated gene 3, that is by X-rays. They took one more Petri plate, and the same procedure was done, and they mutated gene 3 by the X-rays. X-rays, okay, like some bases got deleted, and it could not produce enzyme 3. That means, obviously, the particular neurospora cannot convert the simple medium into arginine. That means the neurospora which cannot synthesize arginine, that is nothing but amino acid. Got it? Arginine. I am talking about another type of amino acid. It is not the codon's amino acid. Okay, it's different. Arginine. Okay, so the neurospora could not synthesize arginine, were called as auxotropes. Please note this point. Auxotropes. What are auxotropes? These are mutant neurospora. Which gene is mutated? Gene 3. Gene 3 could not produce enzyme 3. Because of this mutation, the enzyme 3 was absent and it could not convert citrulline into arginine. So the neurospora could not produce arginine. That's it. So this was the thing, and finally they proved that there is always a single gene always going to produce only one protein. That is nothing but, according to them, protein in the sense enzymes. Okay, so that's all about the beetle and totems, one gene, one enzyme hypothesis. Hope you understood this. I would like to give you some more information regarding different scientists and their work. One scientist called Indra, here let me write Indra. He proposed that one gene will synthesize only one polypeptide chain. So remember, one gene, one polypeptide hypothesis was proposed by a scientist called as Indra. That is enough. Not much explanation is required for it, not necessary to, just remember it. One mRNA will produce only one polypeptide hypothesis. One mRNA, one polypeptide hypothesis was given by Baltimore, a famous scientist, Baltimore and Jacob. Just remember, Baltimore and Jacob proposed one mRNA, one polypeptide hypothesis. Even the Baltimore, just for your general knowledge, he classified the viruses into 6 different classes. 6 classes of virus was given by Baltimore. It is famously called as Baltimore's classification of virus. Okay, in 1970. Anyhow, in your higher classes, you are going to study about Baltimore's classification system of the viruses. Even nowadays, the COVID-19, the virus, coronavirus, belongs to the class, that is virus class 3, which is given by Baltimore. Because of his study of different types of viruses and classified the viruses into 6 different types, it was very easy for the scientist to classify virus, that is coronavirus, now that is in 2020. Fine, not required, just for your general knowledge. I would like to add one more thing. Who called tRNA as the adapter molecule? Adapter molecule. Who called it? Francis Crick. Francis Crick actually, he called the tRNA as the adapter molecule. You know the reason why? The tRNA, which has got complementary bases in its anticodon site, which will read the codons which is present in mRNA. So codons and these anticodons are complementary to each other. Based on this complementarity, the tRNA will bind to the codon and it will leave one amino acid daily in the ribosome. Okay, you will understand when I explain about the translation process. It's very easy. Remember it, adapter molecule, the term given to tRNA by the Francis Crick. Just remember it. That's enough.